The overall goal of the following experiment is to obtain multiple macrophage cultures from mixed primary cultures of rat liver cells without the need for complex equipment or skills. This is achieved first through dissociation of adult rat liver cells by collagenase perfusion, followed by isolation of the parenchymal hepatocyte cell fraction. Next, the cells are incubated in T75 tissue culture flasks with growth medium to establish mixed primary cultures of liver cells. Then, after 10 to 12 days of culture, the culture flasks are shaken to facilitate transfer of the macrophages to plastic dishes, from which they are then harvested by selective adhesion after a short incubation period. Finally, more than 10 to the 6 cells can be harvested repeatedly from the same T75 tissue culture flasks at 2 to 3 day intervals for more than 2 weeks.
Isolation methods for liver macrophages have been well described. However, most of the previous methods require sophisticated equipment such as a centrifugal elucidator and refined technical skills. Here, we present a simple and novel methods to obtain liver macrophages in sufficient number and purity from mixed primary cultures of adult rat liver cells that requires no special equipment or advanced laboratory skills. Animal dissection, perfusion of the liver, and preparation of hepatocytes will be demonstrated by Doctors Noriko Yamanaka and Miyako Yoshioka at the National Institute of Animal Health. Then, isolation and purification of liver macrophages from culture flasks will be shown by Dr. Takato Takenouchi at the National Institute of Agrobiological Sciences. So, let's get started.
To prepare for perfusion of the rat liver, begin by pre-warming 1 bottle of washing solution and 1 bottle of collagenase solution. Then, after confirming sedation by skin pinch, place an anesthetized adult male rat in a stainless steel pan and spray 70% ethanol onto its abdomen and thorax. Next, make a small cut in the middle of the abdomen with dissecting scissors and peel back the skin. Then open the abdomen with the scissors and confirm the location of the portal vein. Next, pass a surgical thread under the portal vein and loosely tighten the thread. Then clamp the distal part of the inferior vena cava and mesenteric vein with a mosquito clamp to prevent blood reflux into the liver. Open the thoracic cavity by cutting the diaphragm with scissors and expose the heart. After making a small incision in the portal vein with ophthalmic scissors, insert a 2 mm plastic catheter into the vein and tighten the thread firmly around the catheter. Load a perfusion system with the pre-warmed washing solution and then connect it to the catheter. Begin perfusion in situ at a rate of 10 milliliters per minute. At the same time, make a cut in the right atrium wall with dissecting scissors. Continue the perfusion for 10 minutes, then switch the perfusion solution to the collagenase solution and perfuse for 10 to 20 minutes at a rate of 10 milliliters per minute. Fill a sterile beaker with 25 milliliters of collagenase solution. Then remove and carefully place the perfused liver into the beaker. Mince the liver into small pieces with scissors. Add 75 milliliters of cold MEM into the resultant cell suspension. After gentle pipetting, filter the suspension through a 100 micrometer cell strainer to remove the connective tissue and the undigested tissue fragments. Collect the filtrate into 50 milliliter conical tubes. Transfer the filtrate into 50 milliliter conical tubes and wash the cells 4 times by first spinning down the cells at 50 Gs for 1 minute at 4 degrees Celsius with the break off. Carefully discard the supernatant and then resuspend the pellet in cold MEM and spin the cells down again. After the last wash, resuspend the liver cells in growth medium. Now seed the cells in 5 to 10 T75 tissue culture flasks at a density of 6.7 times 10 to the 4th cells per square centimeter. Place the culture flasks in an incubator and replace the growth medium every 2 to 3 days. After 1 day of culture, parenchymal hepatocytes spread on the flask's surface and show typical polygonal cobblestone-like morphology with 1 or 2 round nuclei. Within a few days of culture, parenchymal hepatocytes lose their epithelial cell morphology and transform into more flattened fibroblastic cells. Around day 6, phase contrast bright round macrophage-like cells start to proliferate on the fibroblastic cell sheet. The growth of the macrophage-like cells reaches maximum levels around day 12. The number of macrophage-like cells declines around day 19 when the fibroblastic cell sheet starts to degenerate. Thus, around 7 to 10 days of culture, we suspend the macrophages that have proliferated on the cell sheet into the culture medium by reciprocal shaking of the culture flasks for 30 minutes. After the macrophages have been resuspended, plate each of the 100 mm non-tissue culture-grade plastic dishes with the medium from 2 T75 flasks. Refill the culture flasks with growth medium and return them to the incubator. Incubate the plastic dishes for 30 minutes. After the incubation period, wash the dishes 3 times by aspirating the medium and gently rinsing the plastic dish with PBS. As seen here, as early as 10 minutes after plating, macrophage-like cells attach to the dish surface, while other contaminating fibroblastic cells remain suspended. After the PBS rinse, a highly purified macrophage population is obtained. As seen in this figure, the cells gain typical macrophage morphology after 40 minutes of culture, and mitotic cells are frequently observed, as indicated by the arrows. To collect this new highly purified macrophage population, add 1 ml of TRIP-LE Express solution to the washed cells and place the dishes back in the incubator for an additional 10 minutes. After this incubation period, add 9 ml of growth medium and then gently scrape off the attached macrophages with a cell scraper and transfer into a 15 ml conical tube. Centrifuge and discard the supernatant. Add 1 ml of the growth medium. Dissociate the resuspended cell clumps into single cells by vigorous pipetting. Enumerate the cell number by hemocytometer. As shown in this graph, more than 10 to the 6 cells can be harvested from a T75 culture flask repeatedly at 2 to 3 day intervals for more than 2 weeks, enabling a total cell yield of 10 to the 7 per T75 culture flask. As shown in these images, the isolated cells are immunostained with monoclonal antibodies against rat macrophage antigens such as CD68 or ED1 and CD172A or OX41. These cells possessed functional properties of macrophages, such as active phagocytosis of FITC-labeled microbeads.
In this video, we presented a simple and efficient method to obtain macrophages from mixed primary cultures of adult rat liver cells. Our procedure does not require complex equipment or skills, but provides sufficient numbers of pure macrophages that can be repeatedly harvested from the same culture flasks. This method can be applied to other mammalian species. Thank you for watching the video and good luck in your future experiments. Cheers!